This is Gary Miller from Copenhagen, Denmark. I wanted to highlight a manuscript that came out in Nature Medicine, led by Chirag Patel at Harvard, who is one of our multiple principal investigators of the NEXUS project, the network for exposomics in the United States. In this paper, his group looked at over 100,000 associations between exposures and various disease outcomes, really providing this framework of exposome-wide association studies, which is analogous to what we see for genome-wide association studies. And one of the really critical take-home messages here is it's not about individual single exposures. We aren't exposed to one thing at a time. We are classically exposed to a mixture of various compounds. You could think about something if you were eating a diet that had high levels of pesticide residues in it. It's not just a single compound there's multiple pesticide residues. And then there's all the chemicals, the natural chemicals in the plants that tend to be healthy. They're all there at the same time. Or something like air pollution, where you can have thousands of different constituents there. And the idea is you can't just be thinking about one at a time sort of approach. And again, we've seen this for genetics, where they've gone from looking at single gene mutations in a disease to polygenic risk scores, where it's combinations of genes that lead to it. And what Chirag's team showed is that these combinations of environmental exposures and this exposome framework was associated with many of these different outcomes, health and disease outcomes. I think it's really a pioneering paper in the field, and it really sets the tone for many future studies on how to do this large-scale biology. This work was done using NHANES data, which is a CDC-run program where they ran hundreds of different compounds they would test in the blood. And I think that just showing that they had this very highly accurate measurement of thousands of these variables. But with the newer techniques that we're using now with high-resolution mass spectrometry, we can measure tens of thousands of things, if not hundreds of thousands of features in these exposures. And I think if we build upon the framework that Chirag's team showed in the Nature Medicine paper, I think we're really going to kind of open the floodgates of discovery around human disease. So I strongly encourage you to look at the March 18th issue of Nature Medicine. Chirag Patel is the first author on that. I think it's really a fantastic paper, and I encourage you to read it.